Hello, everyone. Welcome to today's Cultivating Success webinar. Today we'll be talking about white rot in garlic, and we have an excellent lineup of presenters here for you today. The Cultivating Success program was originally founded in 2000 as a partnership between the University of Idaho, the Washington State University Food Systems Program, and a nonprofit organization called Rural Roots. So with that, our three presenters today are Jensen, an extension educator from Bonner County. We have Kate Painter with us as well, another extension educator from Boundary County. And then we also have James Woodhall, who is from the Research and Extension Center in Parma. And my name is Mackenzie Lawrence. I'm the Cultivating Success Coordinator, and I will be your moderator today. So with that, I'm going to hand it over to Kate.
Hello. Thank you, Mackenzie, for introducing us and for moderating today. I'm going to talk about a white rot, which is a serious disease of garlic and onions. It does affect all allium crops, but it is garlic and onions are most susceptible. So garlic and onions are an important crop for small farms in North Idaho, as well as a popular home garden crop. In 2018, we discovered white rot in a garlic plot at the Boundary County Community Garden, which we collaborate with the nonprofit that runs the community garden here. In 2019, white rot was reported in both Boundary and Bonner County home gardens, as well as a commercial organic farm in Boundary County. And you can see the pictures here showing the kind of yellowing and wilting of the leaves starting at the base of the plant and going up. And you can see some of the fluffy white fungal mycelium on the plants here, as well as the black sclerotia. So it does affect onions, garlic, shallots, and other allium species, including ornamentals, but it is, again, most common on onions and garlic. It's highly contagious and destructive. It can make the ground unsuitable for any kind of allium production for 20 years, possibly more. It is spread by those tiny black sclerotia that you see in this picture, about the size of a poppy seed. And these are a pathogen structure similar to a seed that forms on the decayed plant tissue. Cool, moist conditions between 50 and 75 degrees are most conducive for spread of the disease. And there are no effective remedies for the home garden. And there are no resistant varieties of either garlic or onions. So first of all, you'll see the leaves beginning to die, exhibiting some yellowing and wilting, with the decay beginning at the base, older leaves dying first, and a fluffy white growth might be seen around the base of the bulb. The small black sclerotia will start to form. And then as the roots begin to rot, the plant can be easily pulled from the ground. In fact, you might first notice that plant seems to be falling out of the ground. It will look a little stunted. There are a number of different diseases that can look like this in some aspects, but that's why you need to make sure what's going on. And we had a number of white rot infestations, as I said, in our county, so we got a hold of UI plant pathologist James Woodhall, and he has developed a DNA soil test for this organism. He came up in our area and did field inspections and gathered samples at various sites. We held a workshop in Boundary County with a number of gardeners and that had reported this, and James actually was able to demonstrate in the classroom his DNA soil test, so that was very exciting. So to help the commercial organic farm in Boundary County, I started a white rot decline trial. It's a randomized complete block design with 5 replications in 20 4x5 meter plots. This area had a severe outbreak. 2 types of cover crops were planted, both mustard and clover. Mustard cover crop was thought that might inhibit this disease. And then we used 2 different rates of onion powder, high and low. Soil samples were collected and a DNA soil test for the white rot organism were conducted in the fall of 2019 and spring of 2020. And then we collected samples in the fall of 2020 as well. And we hope to continue this and see if we can't see some decline. So far, we have not. The three of us wrote a green thumb bulletin called Beware Signs of White Rot in Garlic and Other Allium Crops. So you can easily find that by Googling white rot in garlic at the University of Idaho website. It is a green thumb publication, bulletin 955, as it says in the slide. Okay, Jen, do you want to go next?
Thank you, Kate. I'm just going to quickly talk a little bit about the quarantine rules regarding the allium species here in Idaho. And the Idaho Administrative Code 020605 governs plant diseases and quarantines. And so this affects growers of onions and all alliums within particularly the designated counties. There's 20 designated counties. The overall rule also affects other plant disease and quarantines regarding other species. But we'll just focus on the onion portion and the allium portion, which is that subchapter B, which is there because of this white rot disease. And the general purpose of the rule is to provide regional consistency for plant pest quarantines. And so it establishes regulated pests and products, and then designates these control areas and quarantine areas. And all of the potential problem diseases that are covered under this code can impact the crops and commodities that are grown in Idaho. So that's why they've set up these quarantine requirements. And so the designated counties, these 20 counties that you see listed here, are the ones that are specifically affected by this code. Okay, next slide.
Jen, could I ask you a question before we move on? Ron asked, why is it just these counties of Idaho that have this quarantine?
That's a good question that perhaps might be better directed to ISDA. This is where there is a lot of allium production done, large scale onion and garlic production. So I imagine that's part of it. It's also a potential area where this disease could live. And so, but for more specifics, perhaps addressing the Department of Agriculture, which I have contact information at the end of my slides for that. Thanks. Okay, so this rule, the subchapter here affecting alliums states that no person may import into the designated counties bulbs, sets, or seedlings of onion, garlic, leeks, chives, shallots, or any other allium species, including the ornamental alliums. So you could import seeds, but none of these other propagating methods. And so if you are looking to have these other bulbs or sets within those counties, then the allium must have been produced, those vegetative propagule materials have to be produced from seed within the designated counties. And the only exception to that would be bulb set seedlings of allium species that are actually produced in Malheur County. And if you're looking for a list of those allium producers within that area, you can contact ISDA. I was given a list in 2019. I do not have an updated list at this time though, but they are folks that have stock that's for sale that has been certified for freedom of this onion white rot. And so what the ISDA, part of this rule states that the director is the official term, but that basically means the Idaho State Department of Agriculture. It may inspect any regulated product or product planting area within those designated counties at any time. And so they do, and you can see in some of the plant industry end of year reports. For instance, this is again a little bit older, but in 2018, they inspected 11.6 acres of garlic production, and it was all found to be apparently free from the white rot. They had inspected 944 acres of onion production, and they did find white rot in 12 of those acres, in which case the ISDA will direct the control of the infestation and may seize any of the regulated products from that area. So contact the ISDA for that list of those certified growers there. And my last slide is just some contact information for the Idaho State Department of Agriculture, their phone, and then the email next shows where you can email to get more information regarding this rule, and then also information about any producers within that area that have the certified stock available. Okay, I will go ahead and let James take over because I know he has a lot to share with us.
Thanks for inviting me to talk today. I'm going to focus on the diagnostic and the aspect of the project, as well as talking a bit about the biology of the disease in the first instance. So I'm based at Palmer Research and Extension Center, and it's one of two diagnostic labs. We do research as well, but it's one of two diagnostic labs for plant diseases that the university offers. Both labs cover any plant disease, and we do a wide range of tests from the traditional to molecular testing, particularly of soil and plant material. And both labs are in the National Plant Diagnostic Network, which does provide us with funding, and also a network of laboratories where we can tap into additional expertise if required, or if we need surge capacity, say. So I was really happy that Kate got me involved with dealing with this organism, white rot, because I worked with it in my previous job in the UK. It's one of those organisms which you might see two names for it. Although mycologists are working towards just having one name per fungus, if you dive into the literature, especially anything over 10 years old, you might see an alternative name. It's a particularly aggressive and nasty plant pathogen. It's spread by soil-borne sclerotia, which are pretty small, and just one of those in 2.2 pounds of soil can initiate very, very severe disease. And I've heard anecdotally that even in 20 pounds of soil, disease can be initiated, although it might not be severe. But when I was working on it in the UK, if we just grow up some onion plants in the glasshouse or the controlled environment chamber, we would just drop one sclerotia onto that plant and it would completely wipe out the plant. It was quite phenomenal how aggressive it was. As Jennifer mentioned, it is a regulated pathogen in the Treasure Valley where I'm based in Palmer. And I would argue that this pathogen is one of the most important diseases of alliums globally. So these are what the sclerotia look like. So basically sclerotia are sort of hardened survival structures of the pathogen. They're about a quarter of the size of a ballpoint pen. And essentially it's a soil-borne disease. It kind of infects the bulb from below. Well, it goes into the roots and then can infect the bulb. And if it infects early, it can go up the stem, well, the leaves as well. But typically you get this infection, this hollowing out of the onion bulb. And what actually happens is it literally converts the plant material into sclerotia. So if you were to sort of like pick up this bulb that's shown on this photograph here, just millions of sclerotia would come out. So as somebody who works in a lab in a quarantine area for the disease, it makes me very nervous receiving material like this, even though we're a diagnostic lab and we are there to diagnose anything. We're in a production area where 25% of US onions are actually grown. It kind of makes me nervous to receive these kind of samples. So these soil-borne sclerotia, they're not just prolific in terms of the amount of production on a bulb. They can survive for a long time. At least 20 years has been proven, but anecdotally 30, 40 years is likely. And it can be spread through plant material, as well as movement on soil, equipment, humans, and irrigation can kind of move the sclerotia down as well and spread it throughout a field. So if you do get one onion that's infected, it can really just spread down that field with the flow of water. And this disease, it's a soil-borne disease, so it's not like a foliar disease which can spread quite rapidly. It's a soil-borne disease, so it spreads, over a longer period of time, but it is gradually expanding its range over the Western United States. And it has been found a couple of times in the regulated area of the Treasure Valley, but there's always been some very strict eradication activities, and it's not considered established there. So what can you do if you get it? Well, there are some options if you are in an infested area and you want to try and manage the disease, such as hot water treatment. We are experimenting with Kate in one infested field, looking at some germination stimulants. So just sort of purified extracts of alliums to try and germinate the sclerotia in the absence of a host, and that would hopefully kill the sclerotia. A few fungicides have been reported to have some activity, but in reality, avoidance is the only effective means of disease management. So what do I mean by avoidance? Well, Jennifer explained about quarantines, which is essentially hygiene on a larger regional scale. But if you do have effective crop rotation, putting out your fields out of action, out of allium production for some time is probably the only way. And then there's also soil testing. So in a lot of places where white rot is endemic, people do test the fields as a routine measure. So how do you test the fields? Well, there's two ways of testing the fields. You can kind of sieve the soil samples for sclerotia and try and plate them out. And that's quite laborious, and it can vary quite a lot depending on the skill of the laboratory operator. But we do offer a lab-based molecular test for various soil pathogens, including white rot, the causal agent of white rot. And this does take some time as well. But the advantage of this method is once you've got the DNA, you can test for multiple onion diseases or any disease for that matter that we have a molecular test for. And essentially this is, we sort of mix up the soil with ball bearings and a buffer, which kind of preserves the DNA and sort of ruptures cells as well. And we put that on a paint shaker, which kind of homogenizes it, causes a physical disruption of cells as well. And then we have various spin steps in a benchtop centrifuge to further purify the sample, followed by a final DNA purification on a robot called the Kingfisher. And that leaves us, we go down from like a kilogram of soil down to sort of 200 microliter DNA sample, which we analyze using a method called TACMAN or real-time PCR. TACMAN is a version of real-time PCR. And the reason why it's called that is because the DNA polymerase is kind of looks a bit like a Pac-Man in terms of it kind of binds along, it hits a probe, which is in the reaction, which when it's as a probe, it's not emitting any fluorescence, but when the Pac-Man kind of eats up the probe, it does emit fluorescence. And we can monitor that amount of fluorescence in an assay and work out if DNA is present, our target DNA is present. It's all down to primers, and those primers need to be specific for the disease we're trying to detect. So if those primers are not specific, if those primers do not detect, do not bind to the DNA that we're interested in, because the DNA is not there, you don't get any fluorescence. So essentially, this is what it looks like. If you get a positive reaction, like with the blue line, you get a kind of a nice curve. If there's no DNA of your target there, it's a flat line. And then this CT value, the sort of more it is to the left, the more DNA of our target is there. So we can quantify the amount of disease in a sample using this method. And I actually published this for white rot back in 2012. We tried it on various amounts of soil. You can see we've got a cycle threshold. The more sclerotia in a sample, the lower the cycle threshold. So it is counterintuitive, but it's a method that's published and we know it works. So we use that in responding to this outbreak to try and see how many samples were positive. And we also tried to confirm that Southwest Idaho was still free of the white rot fungus. And you can see the only soils that were positive were a few soils in Boundary County. So the soil testing enabled us to kind of zero in on some infested areas. However, I was very nervous about going into 2019 with the prospect of receiving infested material from bulbs, garlic or onions, which could be just completely infested with these hundreds of thousands of sclerotia. The reason is Palmer is in the regulated area, and although we're a kind of self-contained research station, several fields around the station are actually used for commercial onion production, and in the area, 25% of US onions by weight are grown there. So I really didn't want to be that guy who introduced white rot to the Treasure Valley. So I had to think of a way of minimizing the amount of material that could be submitted to the lab, but we still needed to confirm diagnoses. Although you can visually confirm it, there are symptoms in some cases which are atypical, and as presence of this disease kind of puts out a field out of action for quite a long time, we have to be absolutely confident in this diagnosis. So we looked at point of care testing as a solution. So over the last 10 years, there's been a lot of interest in isothermal DNA methods. So these are kind of portable molecular testing methods. And they literally are, I mean, this one is basically handheld, and it enables you to do a molecular test in the field. And the reason why it does that is that it's isothermal. Unlike TACMAN, which kind of uses heat to separate the DNA strands, the isothermal method, the LAMP method, actually uses an enzyme to unzip the DNA. And unlike TACMAN, where you have 2 primers and a probe, in this case, you actually have 6 primers orientated in such a way that it produces these massive loops of DNA on each round of amplification. So it's much more rapid than a PCR, and it can be field-based as well at a cost of slight sensitivity. Anyway, the other advantage of the isothermal method as well is that it can be used with a very crude DNA extract. So what we do is we can do this for any host. Here we're doing it actually for grapevines with another test we developed. But what we do is we just take some of the plant material, we add it to a 5 ml tube, we shake it up in a buffer, which has got some sodium hydroxide in to give it an alkaline lysis. But there's also a buffer in there which enables it to have a very high pH for the alkaline lysis. But when you dilute it 1 in 10, it takes the pH down to 7 to enable it to be suitable for LAMP. And what we also do is, rather than using pipettes like we would do in the lab, we actually use loops. So each of these loops can take 2 microliters of DNA, and we can use that to transfer in the tubes. It's a little bit easier than using pipettes and tips in the field or on the side of a field. So it's a pretty quick field-based DNA extraction method as opposed to one in the lab, which can take an hour. So we developed a sort of a point of care diagnostic very, very quickly from when we saw the initial outbreaks in late August 2018. We had an AMP assay ready for deployment the following spring. What we did was we sort of aligned the DNA sequences to design this, DNA sequences that were publicly available. And then we designed the LAMP assay primers over mismatches between related species. And then we tested it for specificity and sensitivity. And we did a comparison with the standard test, which was the TACMAN assay I developed. And this is the specificity testing. So we take a panel of isolates that are isolated either from onions or garlic. So we're making sure it's specific for anything that it could encounter on the host of interest. And also we took some related species either from the soil or from potatoes or what have you to make sure that it was specific on a taxonomic basis as well. And thankfully, the assay only worked with the DNA we got from the 2 original samples that we received. So we knew it was specific for white rot. Then we did some sensitivity testing, and we sort of looked at a range of DNA. And as you can see, that the assay is very, very fast. It takes down to picogram or beyond 1 picogram of DNA. No, sorry, 5.6 picograms of DNA. So almost down to the femtogram range. And that reaction, it all can take place within 10 minutes as well. So that's a pretty fast and sensitive reaction. And then what we did was we deployed the assay at a few workshops where Jennifer and Kate invited growers to sort of bring in samples and where we could test it in front of them. And we actually detected the pathogen within about 6 minutes, 6 minutes 30 at both workshops as well. And then we also tried it out with some individual sclerotia that we took off the base of one of the bulbs, and we could detect down to an individual sclerotia as well. So it shows that the assay is very, very sensitive. And this is a picture of one of the workshops. And this is not a picture of me looking very smug. It's a picture of me relieved that it worked. So I was very happy because, you know, when you do one of these tests for real, you never know, even though how much you test it in the lab when you do it in front of an audience. But yeah, they worked very, very well. And it was pretty good to see detection at about 6 minutes, which is a very fast assay indeed. So I think it's pretty amazing that we can do a molecular test and get a result within less than 10 minutes. So to conclude, I think this is it was a good exercise. It showed the rapid development of a point of care molecular test and its deployment as well in workshops. And sort of looking back through the literature, there's been a lot of LAMP assays developed and published, but very few cases where they've been actually deployed in a real life situation. So sometimes they are deployed at a point of entry, say at ports by regulatory authorities. But I think this is one of the first instances where we've actually, a diagnostic lab has deployed a point of care assay remotely to try and avoid material being submitted through a quarantine area as well. The other great thing about doing a LAMP assay, especially when you could do it in front of an audience within 10 minutes, is it does allow for much greater engagement with the growers. You know, they can actually see a positive and connect symptoms in their brain, you know, with the diagnostic result. It really brings home the message, especially when you're dealing with a disease as important as white rot. So I think it's a great tool for that. And in this instance, this LAMP assay is very useful. So as I mentioned earlier on, the Treasure Valley does have the sporadic outbreak of white rot. And this test that we've developed for LAMP will be a very useful confirmation test. Even we should have developed it anyway, because, you know, with the TACMAN and the LAMP assay, we have 2 tests to kind of confirm each other. And the LAMP is a different chemistry. It's a different gene that we're doing. So it's quite a powerful confirmation test, just as part of our quarantine response. And it's so quick, we can get an answer quickly and deploy the eradication measures earlier, because you don't want to take any chances with this pathogen being so important. And finally, I'd just like to acknowledge the team. It was a great team to work with. I really appreciate Kate's energy with this project and bringing me on board with it. I think we've achieved something quite nice with the LAMP assay. And Jennifer organizing the workshop at Sandpoint. And then my crew, Miranda and Lara. And then also I'd like to thank the NPDN and the Idaho Eastern Oregon Onion Committee for providing some cash for the method development used in this project. And thank you for listening.
Thank you so much. That sounds like a really interesting project. We have a couple of questions, so I'll ask them of all of you, and then you can all share your answers, or whoever thinks that they're the best person to respond. Dwayne asked, is there any significant problem in Latah County, or maybe the Latah County area, that any of you are aware of?
It was reported, I think back in 2013. So I didn't have it on my table there, but yes, there is a paper where they do discuss it, and that's publicly available on Plant Health Progress. So if anyone is interested in that, I can send it. But I think in terms of Idaho, there's only really the Treasure Valley, and then there was Latah County, and then until recently it was, Boundary and Bonner County as well.
Awesome. Thank you. Arlene asks, once white rot is diagnosed in an area, does that automatically mean that area can't grow any allium for at least 20 years? So like if it's at your home, are raised beds with fresh soil an option for growing things in an area that had white rot?
It is an option. I think the onion roots can actually get down quite a little bit deeper than you think. So you just have to make sure it's well out the way if you really, really need to grow a susceptible host. Personally, I wouldn't take the risk because you'll just keep on bringing it back.
That's what we did at the community garden is we made tall raised beds. They're 30 inches tall. And we put down landscape fabric weed barrier on that whole area. And then we don't allow any alliums either, just to be double safe.
Okay, but other crops you could grow in those raised beds?
Yeah, so I think ISDA recommend that you don't grow any sort of below ground crops like tubers, where you could be potentially digging up, and spreading the soil that way, which I think is good advice. But yeah, it's such an infectious pathogen. I would err on the side of caution, but I do understand that there are limitations with what people can do.
Absolutely. Nicole had a question about home gardeners getting their soil samples tested. I think you mentioned that folks can send soil samples to the lab in Parma. I don't know, how'd you go about getting your soil sampled?
Yep, so you can look us up online, and all the contact details and submission details are on University of Idaho Plant Diagnostic Services. For the soil testing, we do have to charge, though. It ranges from $50 to $100 depending on the amount of samples that need to be tested. I wouldn't recommend that for home gardeners. It might be an option for like tree diseases. We do a lot of that for verticillium because trees are a little bit more have a little bit more value maybe than onions. So I wouldn't recommend it for onions unless you're going to plan to grow a lot of onions.
James, didn't you say that where you back in England that before growers planted any allium, they would test for the disease presence?
Yeah, so commercial growers would.
Because it's pretty widespread there.
Yes, it is. So yeah, and you'd have to do a grid sampling, and probably every 10 acres you need to get a sample. We're happy to test for homeowners. It just depends on if you think that the value of that test is worth it. I would say if you've got any doubts of white rot being present, just don't grow alliums.
Awesome.
Well, this has been such an eye-opening, in a bad way, experience for me about how careful we need to be with dirt on our shoes or tools, the way we spread it in the community garden. One year I saw it, and I did not know it was white rot. I just thought it was a low-lying wet area and it had some fungal disease or other. And then with the rototiller, we spread it around the garden. especially probably, well, I'm not even sure to what extent it is in the gardens. And so we have signs posted all around the community garden, and it creeps people out that they've got to make sure there's absolutely no dirt attached to anything and just use the garden tools that we provide. And but also the home gardeners that found it are pretty unhappy, you know. So I think spreading awareness through this publication and the workshop and has been really helpful to make people realize sharing plant material is hazardous. And so I think Jen's work showing that you could get that inspected plant material might be the way to avoid that problem, right?
Right. And I was also just going to add to that this quarantine rule does also regulate the movement of equipment from areas where the disease is present into these 20 designated counties. So that equipment should not be moved, or with special approval from ISDA, my understanding is it can, once it has been steam cleaned, to make sure that you are not transporting any of that soil or the pathogen.
So what was quite encouraging when we did the soil testing at I think at 2 of the sites, including the community garden, we tested obviously the allium plots, but then we did test around you know, with other crops in them, and I think we caught some of this quite early on, in that it was only in where alliums were grown we got the positive soil detection. I think it didn't really get into those other areas. So early detection, as we've got available here with the soil test, is critical.
Absolutely. We had another question come in. Dwayne asks, are the commercial bulbs available going to be free of white rot, and how are those regulated?
From my communications with ISDA, the folks that are on the list that they provided me offer they offer the bulbs or cloves or whatever it may be, and it is certified as being free from the onion white rot. I believe, well, and this, I'm not exactly sure, if my understanding may be that they are inspecting the fields and the soil, doing soil tests within that area where those crops are coming from. And they may not always be testing every single clove or bulb that comes out of there. But the rule does state that they can inspect either the product or the planting area for that product to confirm that that white rot's not present.
But it also just was a learning experience. One woman who was in the workshop who you did find white rot in her garlic, I remember thinking that she was being really almost over careful. In a previous year, she came in, she said, should I be planting these garlic cloves? I noticed they had like some little dots on them. And I remember looking at them and saying, well, it doesn't seem too bad, you know. I mean, I can't remember exactly, but she was one that then, and so she dug them up and got rid of them. She said, well, I just decided to get rid of them. I don't know. Anyway, we were talking, she's a neighbor. She dug them up and got rid of them. And then she had white rot. So they probably did have white rot, but even though she dug them up and didn't throw them out, there was probably some sclerotia, so.
Well, I have one more question for you all, and it goes back to testing. Ron asked, where is the second lab that tests? I believe you mentioned Idaho Falls, is that correct?
Yes, Idaho Falls are not geared up to do soil testing for pathogens yet, but they can do nematode testing.
Awesome. And then do you have any idea what percentage of Idaho's commercial property would be affected by white rot or has been?
And no, I think it's limited to, at the moment, to 3 counties, maybe the adjacent counties, but we have no data on those adjacent counties to Latter County. I don't know, Kate, Jennifer, do you have any idea how many homeowners you know were affected?
I'm not sure, you know, I think we worked with 1 or 2 homeowners that were affected. In the process of talking to more of the small scale farmers in the area, I don't think we didn't test any with James that specifically tested positive. I did talk to one grower that thought they may have had it several years ago and just completely stopped planting in that area altogether. They didn't plant any crops in that area going forward, which was partly just a result of them changing their farm plan, regardless of this disease, but also as a safeguard, they thought, well, we'll just not use that anymore.
I was just going to say, we screened 7 operations and home gardens, and I think it was 3 sites that were positive out of that screening. So 3 entire operations were positive. Obviously, they have clean areas.
And we had a couple that came to the workshop that said they definitely had it. They came to the workshop, but they didn't bring anything. And and one of the homeowners that had a positive test, he's a commercial grower, and it was in like a home garden spot on his farm. So that was extremely critical that he learned that, so it didn't spread to his commercial operation.
Yeah, absolutely. I don't see any other questions at this time, but I want to say thank you so much to Jennifer, James, and Kate for a great presentation today about white rot.